So today is the first of a pair of sessions on fluorescence. Today, we're going to talk in general about fluorescence as a measurement and debugging tool, and then talk about using plate readers to measure fluorescence. Next week, we are going to focus on flow cytometry as a measurement mechanism for fluorescence. We're going to start, first of all, by just talking about, in general, fluorescence and absorbance as measurement tools. So, getting started, fluorescent proteins are one of the key debugging outputs that people like to use in synthetic biology. At this point, there are a whole rainbow of different fluorescent proteins out there, and they've been used for a number of the very classic results in synthetic biology. Examples of that includes, over on the right, the band detector from the Weiss lab and the repressilator. If you just want to know what are the possible fluorescences, what plus minus, is a marker here or not, you can get up to around 15 or so different fluorescent proteins measured at the same time. But they tend to slop over into one another's channels a lot. So in practice, if we want to do precision quantification, which is often the case, we can use really only about 3 or 4 fluorescent proteins as probes at a time. Generally, essentially a blue, a green/yellow, a red, and something over in the deep red infrared range. There are a number of different instruments that are the good options for how to measure fluorescence. And I've put the pluses and minuses here. So the sort of most basic thing is a fluorimeter. You put the cuvette into the machine and you measure the fluorescence. And the reason to use a fluorimeter is if a fluorimeter is all that your lab has. Most people, however, will probably at least have access to a plate reader, which essentially gives you high throughput fluorimetry because you get to measure a whole plate worth of wells, typically 96 at a time, rather than just one cuvette at a time. Plate reader is great. You get high throughput. You can look at the evolution of fluorescence and OD, giving you some indication of population at the same time. The dynamic range of how great the brightest and the dimmest that you can distinguish is okay, typically around 2 to 3 logs. But the big minus is that you are measuring the fluorescence of the entire population at once. So you can't distinguish the difference between some cells being very intense and others not so much versus all cells being at a moderate intensity. A flow cytometer gives you the ability to get lots of single cell information and a great dynamic range, lots and lots and lots of individual cells, but it's a very disruptive measurement. We'll talk about that next week. And so you can't do time series with a flow cytometer. So it's very complementary to a plate reader in that way. Finally, microscopy is great. Some microscopes don't give you high throughput. Some microscopes have automation if they're more high end. The real value of a microscope is that it lets you get down to the subcellular level, so you can actually see where fluorescence is. You can see the shape of your cells, and you can see how they're laid out in space. And good microscopes will often let you do a time series. But the microscopes really, they trade spatial resolution for sensitivity. Yeah, individual pixels don't actually have that much difference in their range. So, and the field of view is much more narrow. You can only get a relatively small number of cells at once. So there's pluses and minuses for each of these instruments, and they're often well used in complement to one another. Today, we're going to focus primarily on plate readers. But of course, the thing that matters most is not what would be the best instrument you could possibly use, but the instruments that you actually have and the ones that your instructors may be able to best help you use. It's much better to have an expert who can really help you get the most out of an instrument that's not quite so good over badly using an instrument that you're barely allowed to touch. Well, let's talk about plate readers. Let's talk about how a plate reader works. Over on the right here, we have a picture of a typical plate reader. Where you basically put the 96 well plate into a little tray. It sucks into the machine, and then the machine's going to move this around, or in some cases, just have enough material to enough illuminations and reading to read it all directly. But what it's going to do is each of these wells is going to get illuminated by a laser or other excitation light. It's going to shine through the well, and then what comes out of the well is going to be some mix of less of the original color, because some of it gets absorbed on the way through, and more of new colors that have been generated as the cells and other stuff in the well fluoresces. So that new light that comes out will typically get split to go through filters into two different detectors. One that's being used to look at the new light that has been added and quantify the amount of fluorescence that you have. And one that's going to look at the original light and how much is left to quantify how much absorbance, and therefore help you estimate how many cells were in that well. Let's talk about absorbance first. The basic idea of absorbance is that we have a light on one side, a detector on the other, and the light that is passing in between, when it hits cells, it gets deflected. So not all of the light that starts out going through ends up actually making it all the way through. The measure that we get, OD, optical density, a physicist will argue that that's possibly the wrong name for it, but that's the one that we typically use in biology. OD is the log 10 of the amount at the source divided by the amount at the detector. And at a low level, this is actually, this number is actually linear in the amount of particles in between because of the way that scattering impacts. The actual fraction of light that's detected, however, is complex. It depends on not just how dense the particles are, but how opaque they are and how far the light is traveling, because you're going to lose some just by the travel itself, as well as the same density, twice as much is going to remove twice as much. So this estimate is trying to understand something about how dense the material is, even though you may be going for a different distance in it. The amount of scattering that you have typically depends on the frequency that you're dealing with as well. And here, the most typical measurement is at 600 nanometers. If you're using red, you might want to change that to 660 nanometers, because 600 can end up, if you've got a very intense fluorescence, it can end up getting absorbed by your fluorescent particle as well. But it's a good starting point. Fluorescence has a related mechanism, except that we also have light being generated. So light comes in, it hits a fluorescent molecule, and fluorescent molecules all essentially resonate to particular frequencies. And they get kicked up into a higher energy state. How hard they get kicked up depends on the frequency of the light that comes in. And once they're in that higher energy state, no matter how they got there, they then fall out of that higher energy state in the same pattern of emissions, which crosses a whole bunch of different lower frequencies. So it may come in very narrow, even at a laser, and then it comes out smeared across a whole bunch of different frequencies. Here on the right-hand side, you see the most typically used GFP in iGEM, GFP MUT3, which is very, very widely used because it's BioBrick E0040. You can see that this range here is going all the way from about 500 nanometers, which is in sort of the intense green region, out through 580, which is starting to get more towards the red side. This is a fantastic signal to work with from inspecting what's going on inside of your cells. It's extremely sensitive. It's closely proportional to the number of molecules, very well behaved, except the exact unit relationship is extremely sensitive to context and hard to calculate from first principles. In order to figure out how the signal you get relates to the number of fluorescent molecules, we basically always will need to calibrate our measurements. Let me say a little bit more about why that is. There's a lot of different factors that affect the fluorescence measurements that we make. The particular molecule that we use affects it. Every different type of molecule has a different excitation spectrum, that is, how strongly it responds to different frequencies of light, and a different emission spectrum, that is, the mix of light that comes out of it. Different molecules are more or less efficient in converting incoming light to outcoming light. This is something known as brightness or quantum yield. If the molecule doesn't fold right, typically these are fairly chunky proteins. If that protein doesn't fold right, then it won't emit right. And similarly, if that molecule is bound to another molecule, then it may or may not lose some of its efficacy as well. It may get what's called quenched. Things can be going on in your sample. These molecules respond differently at different pH. A lot of them are dependent on oxygen in order to fold properly and fluoresce. Also, not only is fluorescence coming out of your fluorescence molecules, it's coming out of the media. Some media are very fluorescent, some media not so much. And it may come out of the cell, and it may vary with cell state. For example, a lot of cells turn more red as they get stressed and unhealthy and start dying. So they'll start fluorescing red. Those spectrums are big and broad. So if you're using more than one fluorescent molecule, their spectrums can overlap, and we need to disentangle that. And because with a plate reader at least, shining through the sample, if you have more sample in the path, then you may have more fluorescence appearing. Similarly, your instrument itself, the strength of your light can often be varied, the frequency, what filters are in the emission, etc. All of this basically says, in order to effectively interpret what a fluorescence number means, we absolutely have to use some sort of independent calibrant, which we'll show how to do later on. Let me talk about one more key thing before we do a pause for questions, which is these excitation and emission spectra. Here's an example of excitation and emission spectra with the tag BFP blue molecule. So over on the left, this purple is tag BFP's excitation. This is how efficient a particular frequency of light is in stimulating emission. So in this case, we can see, for example, that about 400 nanometers, that's a nice violet light, is the maximum efficiency for stimulating tag BFP. And if we go to more and more on the violet side, to lower nanometers, the efficacy drops off. And by the time we get down to 350 nanometers, we're barely stimulating this molecule at all. Same way as we go from violet down towards blue, our ability to stimulate the molecule starts to drop. On the other side, this blue one is showing its emission spectrum. So if we hit it anywhere in this purple spectrum, we're going to get a distribution of the blue spectrum. And the closer that we're hitting it to 400, the higher that this is going to be proportionally. It's going to scale upwards. Now, let's see what happens when we start thinking about multiple of these. Here is an example of 3 of the recommended proteins that we suggest overlapping one another to some degree or other. Here's tag BFP, the blue one, GFP mut3 over in the green-yellow space, and M-cherry over in the red space. Each of these, we've got the excitation is on the left, the emission is on the right. You're almost always exciting with a higher energy that is shorter wavelength light, and then having it spill out, having lost some of its energy across a lower spectrum. And in this case, these are well separated, because if I stimulate in this purple region, that purple region is separate from what will stimulate GFP mut3, and it has only a little tiny bit of overlap with what will stimulate M-cherry. There's a little bit of overlap there, but the emission is very well separated. So if I have a filter that's looking for blue, it's not going to catch any red. If I have a filter that's looking for red, it's not going to catch any blue, and so on. So this is an example of 3 nicely separated pieces. Whenever you're thinking about using more than one fluorescent protein, you want to think about how to make sure that they are nicely separated like this. Let's talk a little bit more about this. Here's an example of 2 things that are not well separated. This is tag BFP again, and this sort of double humped excitation and double humped emission is M cerulean. These are distinct enough that if we want to say, just be asking, is there an antibody tagged with one of these on something, we could probably do that. We could probably do some plus minus. But you can see there's, throughout the whole range of stimulating tag BFP, we're also going to be putting a bunch of stimulation into M cerulean. And throughout the whole range of looking at what comes out of tag BFP, we're going to be seeing a bunch of energy coming out of M cerulean and vice versa. So it's going to be very hard to quantify a system in which we want to measure one of these being bright and the other one being not so bright. We can fix this by choosing things that separate either the excitation or the emission. The most typical thing that we do is to separate by excitation. GFP mut3 and tag BFP just don't have anything in common in their excitation. So if you hit this with a narrow beam of light over here, you'll get no green. If you hit this with a narrow beam of light over here, you'll get no blue. This is the most typical, and what we most strongly recommend is separating by excitation when possible. If for some reason you can't separate by excitation, for example, you might have a machine that can only do one light source. It can sometimes be possible to separate by emission. Here's an example of that with tag BFP and a molecule called M-ametrin that is stimulated in almost exactly the same range, but emits at a much lower yellow-green range. So you could drop a filter to pull out the blue over here and have that be entirely separate from a filter that's pulling out the yellow-green over here. I'll end by just taking a recommendation. I'm not going to go through all the detail here, but the measurement committee has recommended a set of standard fluorescent proteins and calibrant dyes that we can use with each of them. These are based on both experience, things that people have found very easy to use and fairly comfortable, and also their frequencies, brightness, maturation time, and low sensitivity to pH changes. So this is something that you can go and look up afterwards if you want. There's information on the measurement committee page. If you need to go out beyond these proteins, there's a wonderful resource that I've been pulling up these charts from called fpbaseorg. This is a great place to go and explore what is the whole bestiary of possible fluorescent proteins out there. So let me pause here at the beginning of this first section, and I'll go check where our questions are. In summary, fluorescence can be a very valuable tool for debugging biological systems. And the different instruments we have for measuring fluorescence are good for different purposes. Plate readers are really good for tracing time series. Flow cytometers are good for looking at the behavior of individual cells in population. And microscopes are good for understanding the spatial arrangement of cells and subcellular structure. With a plate reader, you typically get to measure absorbance, also often referred to as OD, and fluorescence. Fluorescence is affected by a lot of different factors, so you've got to calibrate your measurements in order to understand what the fluorescence is telling you. And your fluorescent proteins should be chosen to match the calibrants that you have available and to not interfere with each other.